Uh, can you all see the whiteboard? And can you all hear me?
Yes, sir.
Yes, yes.
I mean, you know that in general, I can draw a graph in bioprocess logic by this kind of very. I mean, I can draw. I mean, I don't. You may not have been taught like this, but if I draw something like QP by QP max, so sort of so QP max and divided by mu, and here we write mu by mu max, which is therefore, I mean, I've scaled it from 0 to 1 and 0 to 1 respectively. Then a perfectly growth associated function would be a straight line at 45 degrees. Do you follow this argument? Because if QP is proportional to mu, the proportionality constant with scaled QP to QP max and mu by mu max will simply become 1, right? I scaled it up. And so this graph essentially represents, and this graph represents what is called growth associated product formation kinetics. Growth associated product formation. And almost all enzymes follow this logic. Growth associated product formation. And therefore, you need to push mu higher and higher values to get higher and higher QP. Because remember, QP is nothing but 1 upon x dp by dt. And of course, mu is nothing but specific growth rate, which is 1 upon x dx by dt. By definition. So this is growth associated product formation kinetics. And for example, let me say for secondary metabolites, etc., there is almost no product formation at 0, but it sort of increases. Let me sort of put it in a different color. It increases like very fast at low growth rates, and then it is almost maximum at this growth rate, and then it tends to fall like this, because high growth rates, or rather, I mean, I made it deliberately like this, it falls much earlier, because at high growth rates, only primary metabolites are formed, secondary metabolites are not formed. So secondary metabolites would look something like this. So you can, as a matter of fact, secondary metabolites look something like this. So as a matter of fact, you can think of various graphs, how they would look like. But my point is that I could think of graphs like this essentially, and this, and this, and this, and so on and so forth. Now it turns out, sorry, that's very poorly drawn, but I hope you understand. So let me to wrap it up very quickly. It turns out that most recombinant proteins follow this graph. What it really means is that as you reduce this, the growth rate to say half, your QP falls to more than half. So if you reduce it to by 20-30%, you notice that QP has fallen to less than half. So you want to keep pushing up mu, because your product formation is actually not just proportional to mu, but possibly proportional to mu square or something. You can see it's y is equal to x square kind of thing. Or mu restore of some power n, where n is greater than 1. And therefore, the tendency for any fermentation strategy, wherever you are trying to produce product which is recombinant proteins, is to try to make mu the maximum possible, mu as close to mu max as possible, so that you can get high productivity. And that sort of requires that oxygen supply, etc., can become quite rate limiting, because you have now the dual problem of having high cell density, because you want high productivity, as well as you want high cell density as well as high mu. And so that then leaves you with this contradiction that therefore mu into x and mu into x are both high, and mu into x is nothing but dx by dt, which is of course proportional to KLAC star minus c. So therefore, or the OTR or the OUR is proportional to OTR, is proportional to OUR, and hence which is equal to OTR. And so oxygen transfer, etc., mixing, all kinds of problems come up, primarily because you want both high mu as well as high cell density to make recombinant proteins. So recombinant protein production, that is the main issue. So that is what I wanted to tell you. I didn't want to get into the bioprocess part too much, but you need to be aware of this fundamental problem. So this is clear to all of you from the problem that we solved? Hello?
Yes,
Yeah?
clear.
Yeah, so it's clear. Now, let me
Yes,
ask
sir.
you a very simple question. Why do you think this is so? Why do you think QP is proportional to mu square or something? Why? Why do you think that the rate of product formation of recombinant proteins is actually a function of, is a graph like this? What is the physiological or biochemistry explanation behind the reason of QP versus mu plot looking like a, why not this? Why not this graph? As a matter of fact, the true graph is more complicated. It is something like this. It is something like this. Sorry, it is something like this. And then, of course, at the higher values, it falls. It falls because of acetate formation. But if you consider this part, the first half of the graph, why is it that it increases? You consider this part. Of course, later part, it falls for another reason. That is because of acetate overflow mechanism, which I talked about, and the formation of acetate. But if you forget this part, which has a clear biochemistry explanation, acetate formation, what is the explanation for the first half where it increases, not just linearly, but in a sort of, in an x square fashion? Can you give me an explanation? From the logic of biochemistry? Any options? We talked about it. We talked about how proteins are produced. What is the protein synthesis machinery? What is the rate limiting step? Go on. Guesses? Why this graph? Why do engineers get this graph? What is the connection with the molecular biologists? So as I was telling you, come on, some answers. So as I was telling you, and since you're not volunteering, that DNA to RNA, mRNA, this is what is transcriptional control, and as I said, promoters etc. can be very strong, which ensure that this is no longer the rate limiting step. And I told about the fact that mRNA levels do not go up in steady state so much as you force the promoters to become stronger and stronger. So this is no longer the rate limiting step, and the rate limiting step really becomes this to polypeptide, which is the translation step. And this translation step requires what? Energy and ribosomal machinery synthesis and all these things. And all these translation steps are directly related to mu. The translation rate is proportional to mu. And this function, mind you have heard of this, I'm sure you would have heard of this, in any structured modeling. I mean, we're not getting into structured modeling, but in any structured modeling, the amount or the level of rRNA in the cell, the levels of rRNA levels in the cell, these levels are a function of mu, which means that the level of rRNA, I mean, I'm putting some random numbers, maybe 15% when mu is 0.2, 15 is too high, 5%, and will become 7% when mu is 0.3, and become 8% when mu is 0.4 or something like that. Which really means that as mu increases, QP increases because it is proportional to mu, as well as per unit cell it is higher. So actually YP by X is actually increasing. Notice that if YP by X was constant, QP is proportional to mu. QP is proportional to mu when YP by X is a constant, growth associated. But if YP by X is increasing, so sometimes we use this term, it is more than growth associated, which is a silly term, but tries to capture the argument that as we increase translation rates, and as we increase mu, QP increases even faster, because it is the kind of function QP is equal to mu square, or something like that, or 1.2 or whatever, it's sort of n. So it is actually, as we increase mu, we get higher and higher productivities, and therefore we try to push it as high as possible, so as to ensure that you get good productivity. And that is the reason why we induce at log phase. And so we have to maintain log phase growth at high cell density culture, which becomes a problem. So shall we finish it here and not talk more about it unless you have any questions? Okay. Now,
Yes.
I think we should wrap up and get on to enzyme kinetics. So today is the last class. 26th tomorrow is a holiday. From Friday is the next class, and Saturday will also be a class to compensate for the 26th. Let us sort of wrap it up with, just for sake of completion, I think a little bit of molecular biology is important. So I really don't want to teach it, but for the sake of completion, we need to polypeptide. This has to fold into the proper protein. And then this protein has to be localized. It can be localized in the cytoplasm itself. It can be localized into the periplasm, and in rare cases it can become extracellular. And so this and all these processes are also mediated by for example, degradation processes that are also taking place. It can get degraded, it can fold, it can misfold. So every step there is a problem. Every step there is a problem. So really the point that I'm trying to make here is that this is really molecular biology, most of it, but I want to complete this description unless you are already aware of all this. Okay. Some response would be nice, but yeah. So really the polypeptide that is made. So we are now not only interested in making the polypeptide, the transcription translation steps, but the further steps, which are all these, have to be carefully controlled because that ensures the quality of the product and the downstream steps, the purification steps. The downstream steps would be significantly different for proteins which are localized in the cytoplasm or the periplasm or the extracellular medium. They would be different if they misfold because then you have to refold them properly again and so on and so forth. And you have to prevent degradation. And so all these steps to how to ensure that the quality is maintained and the downstream processing is simpler is part of the process of production of recombinant proteins. Because finally you are interested in the product. And we have talked about purification methods in the lab practicals, so we will not talk about those details, but we will talk about what are the host cell characteristics and what kind of modifications can you carry out in the host cell and the production methodology so that these things are ensured. So let us begin and say that the first step for the polypeptide to fold is the fact that normally, and biophysicists have spent literally thousands of manuscripts and thousands of people have worked on this. Normally proteins fold correctly, almost always inside the cell proteins fold correctly, but it turns out that the moment you produce a recombinant protein in large quantities, you may have incorrect folding. And when they fold incorrectly, and the most common reason is the cysteine residues, which form a disulfide linkage, SS bond, the disulfide linkage. You are familiar with all these terms?
Yes, sir.
Yes, sir.
So the disulfide linkages, and there are certain very ballpark figures. For example, if you have one disulfide linkage in the protein, most probably it will form correctly. But you have 4 disulfide bridges or 3 disulfide bridges or even 2, you have the probability of disulfide bridges forming incorrectly increases, especially if it is a random process. Think about it. 2 cysteines they have to form, but 4 cysteines, and you can. This is what you want. This is what you want, cysteine, and these are the correct bonds, but you may well have this and this, or this and this. All these are incorrect bonds. Right? So there are higher probabilities of incorrect formation, especially if the fold is not correct. Because after all, the protein folds in a certain way, bringing the cysteines closer to each other, thereby naturally allowing the bond to form. But if they are not close together because the fold has not taken place properly, and then your cysteine bridges will not form correctly. So you could have incorrect disulfide bridge formation, disulfide linkage formation. And so proteins which, but the question really is, how do proteins fold correctly normally? And normally is the key phrase. Why don't they fold correctly when recombinant proteins are produced? And the answer obviously is twofold. One, of course, is that it is a foreign protein. Maybe it is not able to do it somehow. It is in a foreign environment, but that's a vague answer. But the more correct answer is that they are being produced in very large quantities. And the rate of production itself is what is the major issue. So it is the rate of production which actually, so your recombinant protein, is being produced at a very fast rate. Now, I mean, rate of production is high. And what we have is a folding process which is competing with the rate of production. Now, folding is of two kinds. Protein folds. Why do proteins fold correctly? That question is very obvious, I thought. Why do polypeptides, why do nascent polypeptides, why does the nascent polypeptide attain its proper shape of an active protein? This answer at least all of you should know.
So they look to minimize their energy rate.
Delta G minimization. And this process, have you read something in biophysics about hydrophobic collapse and molten globule, stuff like that? Have you heard such terms?
Not really.
No, sir.
So molten globule state, which is really an intermediate stage when the peptide is folding, you can see that the word molten globule sounds very, very, very logical. The molten globule state is an intermediate stage, and there is a hydrophobic collapse, which means also that hydrophobic core, the hydrophobic core of what is hydrophobic collapse? Again, that makes a lot of sense, right? Sounds logical. That all amino acids which are essentially hydrophobic would try to go into the core of the protein when it folds. They would like to hide from water. The hydrophilic residues will go outside, and so there is a hydrophobic collapse, which is essentially proteins, the amino acids wanting to rush inside and get the hydrophilic, get away from water, and the hydrophilic molecules being attracted by water, and so there is a natural tendency to fold correctly. So, and in intermediate stages like molten globule, etc., are proposed, and biophysicists have done a very huge amount of work on this, and very interesting. But this is not our subject here. But the folding process and how it works, what are the intermediate stages, what is the potential well that is generated with delta G, right? What is the potential? And you can see very beautiful papers of the potential well being generated. Looks like just like your physics black holes kind of stuff, with arrows going down, and so the protein collapsing into the appropriate stable state, which also is because of delta G. The formation of disulfide bonds then actually further stabilizes the protein structure, the 3D structure. So the nascent polypeptide does not have a 3D structure. The 3D structure stabilizes the protein. Now, recombinant rate of protein production is high, and what really is the most critical, among the most critical factors which causes misfolding, is what is called intermolecular interactions. Oh, I'm touching the wrong place, sorry. Intermolecular interactions. See, under normal situations, the protein is being produced slowly, and there is the one thing that physicists don't talk about, that this folding process, there is a kinetics associated with folding. And what it means really is that the protein folding process is actually slow for some proteins, fast for some other proteins, and each protein has a specific rate kinetics associated with folding, which means it from the nascent polypeptide to the actual hydrophobic collapse and molten globule and then final protein 3D protein structure, whatever is the 3D protein structure. This takes microseconds or seconds or minutes, not minutes really. 3D protein, stable 3D protein structure, it takes a few seconds. But inside the cell, you have the next nascent polypeptide coming up, as the first one is already has half folded. So the hydrophobic collapse has not taken place. It is still in the molten globule form, which means it is half folded. And this new nascent polypeptide comes up. And what does happen? Instead of this fellow being allowed to fold peacefully in its own time, this is now starts interacting with the new nascent polypeptide. And there might be a hydrophobic core here, a hydrophobic amino acid here, and there might be a hydrophobic amino acid which has not yet entered the hole. And they feel that if they interact, then suddenly they can get rid of water. So there is an interaction with partially folded proteins because the kinetics associated with protein folding is slow and the rate of protein production is high. And this interaction of partially folded proteins, their hydrophobic residues, causes them to mix up together to hide the hydrophobic residues, because that is what would be called a partial potential well. You've heard of this kind of things? So they get stuck in this partial potential well, which is lower, and then they have to go up actually and go down. But this is the real steady state, and you can actually reach a steady state, which is a potential well, where the delta G has declined, and it has to now go up again because you have to break this and then bring it back before it goes to the actual minima. So you have local minimas in protein folding, and these local minimas are equally stable, and this local minima then is enforced by the intermolecular interaction rather than the intramolecular interaction. And this is purely to do with the kinetics of folding and the kinetics of rate of production. So you have therefore three solutions to this problem to prevent this interaction, because this interaction then leads to many molecules coming together and they form something which I think Arunangshu would have told you, the formation of inclusion bodies. Have you heard this term? IB formation? Yes, sir. So IB formation is essentially a competition between rate of production and kinetics of folding, with intermolecular interactions dominating the intramolecular interactions. The intermolecular interactions dominate over the intramolecular interactions, then this reaction will dominate. This internal dynamics will become less important, and you will have an inclusion body formation, which is essentially a refractile body, which is completely misfolded, and then this sequesters inside the cell, and you can actually sequestered molecules can be seen. So now how do you solve this problem? There are only 2-3 ways to solve this. One is that you should actually, how do you solve this? Very simple. One, slow down product formation rates. Slow down the rate of recombinant protein synthesis, slow down recombinant protein synthesis. Okay, which ensures that the kinetics of folding protein synthesis is the one solution, which is that if it is slow, then the next molecule is not produced until the first molecule has already folded. And B, somehow prevent interaction. And the most simple ways of preventing interactions are twofold. Prevent this interaction, one, and these are called, this prevention of interaction is achieved by what? You must be familiar with this. Any answers? How do you prevent interaction? Yes. Go ahead. Any guesses? Have you heard of a protein called GRO-ES or GRO-EL, or let me call it GRO-ESL, or DNA-K, DNA-J?
No, sir. No, sir.
So, you've not heard of chaperones? Chaperones are molecules. We have heard of chaperones, yeah. Yeah. So chaperones, HSP60, HSP90, these are heat shock proteins which are generated with heat shock. Heat shock, again, same problem. Anyway, come to that. So chaperones are molecules which work by a variety of mechanisms. They all work in different ways. But let me talk about GRO-ESL, which is the simplest way. The GRO-ESL molecule is a large molecule which comes like this and captures the DNA protein while it is folding inside. So it gives it time to fold by itself, and if and so this next molecule which is coming in and trying to interact this interaction is prevented. So this interaction is prevented because this first molecule is trapped inside by the chaperone. The chaperone provides a safe place, non-interactive environment to the cell, to the protein, for the protein to fold peacefully. And once it is folded properly, then it ejects this and takes the next one. And so it is like a home provided to the proteins for easy folding and allowing them to fold and not allowing them to interact. So you actually, chaperones prevent intermolecular interactions and give the protein space to fold. There are other ways. Chaperones also carry it to the periplasmic space where there is a proper oxidate reduction environment which allows good disulfide linkage formation. They promote disulfide linkages, DNA, DNAK, DNAJ. HSP is a heat shock protein which prevents protein degradation in exactly the same manner by actually entrapping the protein inside a structure. So it's a heat shock protein, and this is all molecular biology which you will learn later on, but I thought a very brief introduction to this. These are all large chapters in molecular biology. And so heat shock proteins do the same thing. They prevent degradation by encapsulating the protein and preventing it from denaturing even when temperatures are high. Similarly, grow year cell and all actually do the same job for a different set of proteins. They work on nascent polypeptides. They capture nascent polypeptides and provide them a microenvironment by encapsulating them and allowing them to fold properly. So this is how, therefore, engineers and biologists have actually co-produced or co-expressed chaperones and shown that chaperones, what is the meaning of the word chaperones in English? I hope you know. Hello?
Like someone who is there to maintain order, or
who accompanies you to keep you safe.
Yes, sir.
I mean, in ancient times, not in ancient times, in medieval times, ladies were chaperoned to the various places that they used to go because they were supposed to be protected and all that. Those silly traditions have fortunately disappeared. And so chaperone is now capturing this protein, allowing it to fold. So that is how the word chaperone has come about. So if you co-express chaperones, you can actually see that from inclusion bodies which are insoluble, you had asked that question, and you get usually soluble proteins because IBs do not form. So to get soluble protein, there are two methods. A, slow down the rate of synthesis. How would you slow down recombinant protein synthesis? Very quickly by engineering methods. Come on, these are common sense questions. Please go ahead, think.
By weak promoters.
By weak promoters, that is one thing, but you already have a promoter. वो promoter लगाने के बाद आपको पता चला कि ये मामला गड़बड़ हो रहा है. तो if you go back to the molecular biologist every time कि आप weak promoter लगाओ तो गुस्सा हो जाएगा. Promoter आपको दे दिया गया है, अब आप क्या करेंगे?
So we can control the induction of the promoter.
Yes, so one thing is that we can reduce the amount of inducer. And as I said, if it is a titratable promoter, reducing the amount, reduce inducer amount, reduce inducer, maybe you can reduce the induction amount. That is one option, but many of these promoters are horrible. They are 0-1, on-off promoters. So really, they are not titratable. या तो ऑफ रहते हैं बहुत कम इंड्यूसर में और फट से ऑन हो जाते हैं. So the response, you know, like control theory तुमने पढ़ा होगा. If you have a response like this of the actuator, then you can never do control, good quality control. When you have a response like this, then you have good control, because you know you can actually titrate and decide किस level पे रखना है, on कितना रखना है, 50% on, 50% off, 20% on, 80% off, by changing levels of inducer. So if it is a responsive titratable promoter, then of course the red line is there. But if it is an on-off promoter, then this methodology does not work. Can you think of a third, even simpler strategy? So A, change the promoter. B, reduce the amount of inducer. And C, reduce temperature. That's the most logical thing, right? Reduce temperature, because temperature reduction will slow down cell growth, slow down protein production, slow down everything. Because everything is finally dependent on temperature, RNA's dependence. And so proteins are sometimes expressed at 37, then at 30, and then at 18 or 25, and so on and so forth. You can reduce temperatures continuously, 25, 18, and so on and so forth, till you get soluble protein by this very fact of slow rate of protein production. Of course, slow growth, slow protein production, slow everything. But at least proteins become soluble. So these are methods of, yes, go ahead.
So it be disadvantageous to slow down the protein production on a large scale industry?
No, it is a problem. It is obviously a problem. Usually, as a matter of fact, because you don't want to waste too much time in growing it, you grow it at 37. And just before induction, you have to reduce it to 18 degrees. Now, unfortunately, this reducing fermentation temperatures down is an expensive and irritating process. But because recombinant proteins, in terms of value for money, they are far more expensive than normal products. Okay. I mean, they are pretty expensive per kilogram or per gram. Actually, per kilogram would be like billions of rupees. So therefore, since they are usually sold in small quantities and the fermenter sizes are therefore never exceed. I mean, there are recombinant proteins being produced in 10,000 liter reactors. I remember at least, no, there are 20,000 tak hai. But 20,000 liters se zyada maine abhi tak reactors nahi dekhe all over the world for recombinant proteins. And that too for industrial enzymes. Most recombinant proteins have biological roles, diagnostics, therapeutics, etc., etc. So they are usually 500,000 liter reactors, and their cost is tremendously high. Most of the guys who are running 1,000 liter reactors abroad, the big guys, per fermenter output is $1 million. Per fermenter, the product price is about $1 million plus. Therefore, the cost associated with lowering temperature is actually a negligible component. If it were a low cost product, something which is, $5 a kilogram or something, but proteins are never $5 a kilogram. They're more like $500,000 a kilogram because they're $5 a milligram. Okay, so therefore, the cost of temperature reduction is usually not factored into the production costs. Especially if you can get folded protein, because if you get inclusion bodies, then you are in huge trouble, because then you have to do IBs. IBs also are handled. I mean, industry does have to handle it sometimes. IBs have to be then unfolded. They have to be unfolded using harsh chemicals and then refolded slowly in large tanks. And I'm not going to get into that. That is part of something else. Let us not get into too much of detail. So they have to be unfolded and then refolded, and that is the only way we can. Just give me 1 minute. Hi, I'm taking a class. I'll call you back. So this is not a very good strategy. But sometimes it is forced upon you, unfolding and refolding of IBs with associated costs. And there are techniques and strategies. Let us not get into that. You will possibly hear about it in either separations or, yeah, go ahead.
In case of decreasing temperature, at which step the protein production is being regulated?
At all steps. Everything goes down, everything, transcription, translation, everything is a function, growth, ATP generation, everything is a function of temperature, isn't it? इसीलिए सांप जो है विंटर में नहीं निकलता है सांप, because all his body functions slow down. They are cold-blooded animals, and one of the biggest advantages that human beings and all warm-blooded mammals have is because they regulate their internal temperature at 37 degrees centigrade. So cold-blooded animals just cannot do anything. Everything is a function of temperature finally. Think about it. Yeah, go ahead.
Sir, protein folding will not be affected because of low temperature?
No, protein folding itself, the molecule, the folding may slow down slightly, but the rate of production will slow down significantly more. थोड़ा स्लो हो जाएगा क्योंकि सिंगल मॉलिक्यूल का भी मूवमेंट इज डिटरमिन्ड बाय काइनेटिक्स, सो ऑब्वियसली देयर विल बी अ स्लाइट रिडक्शन एट द रेट ऑफ फोल्डिंग. 1 सेकंड या 2 सेकंड लगेगा, लेकिन आपका प्रोटीन प्रोडक्शन सप्लाई पर सेकंड एक मॉलिक्यूल आ रहा था, अब 1 मिनट में 1 मॉलिक्यूल आएगा या 30 सेकंड में 1 मॉलिक्यूल आएगा इनसाइड पर सेल. देयर आर बिलियंस ऑफ सेल्स ऑफ कोर्स.
Okay.
So
Okay.
the impact on folding rates is significantly less or negligible compared to the impact on the expression rates. Okay. So shall we, I mean, and so that is how you sort of ensure that proteins are properly folded. Now, very quickly to the other two sections. And because I don't want to talk about it anymore, as I said, this is becoming too much of molecular biology for my liking. I am supposed to be teaching you engineering. I'm supposed to be an engineer, and I'm getting into too much of. So the second part is actually export. And there are two components to export. One is after it, from the cytoplasm to the periplasm. You're all familiar of cytoplasm, periplasm? Yes,
Yes, sir.
no? And then the
Yes, sir.
second and the more and this is more difficult, periplasm to the extracellular space, completely outside. Now, why? I mean, you will say, why not put it out directly outside? But now, unfortunately, it is too difficult. Okay, it is too difficult. This is a more achievable process, and normally it is achieved by putting signal peptides. You have heard of these things? I'm just sort of signal molecules, signal peptides. You have heard of signal peptides which
Yes, sir.
translocate the protein. This is the cell and this is the periplasmic space. And if you have a signal peptide, the signal peptide targets the protein to the periplasm. Targets the protein, and in the process it gets cleaved. And so there is a lot of research which has been done on efficient signal peptides. And people have found many interesting signal peptides, the most common being PELB, UMPE, and so on and so forth. UMPEF, or I don't know, UMPE, I don't remember also. Okay, so these are names of signal peptides, which are small peptide molecules which are attached to the N-terminal end of the protein. And usually these peptides ensure that the protein moves from the cytoplasm to the periplasm, and when it moves, it sort of just gets cleaved off. So the advantage of cleavage, the advantage of having a signal peptide and pushing it to the periplasm is not only that it gets cleaved. So usually, and now I'm getting into more details, but usually you have to start with a ATG. The first step of, and here also there's an ATG. And maybe your actual mature protein does not have an ATG. The mature protein. So now you can get rid of this ATG because you no longer need the ATG here, and this gets cleaved. So your mature protein has the exact amino acid sequence with no extra ATG attached. So this ATG otherwise becomes extra for recombinant proteins. Now your mature protein, if it is being targeted, it will not have ATG, so your quality is better. Secondly, periplasm, of course, gives a much cleaner environment, very few proteins, so your purification is better. And because there are few proteins, folding is much better, and also the appropriate oxidative environment is there, and so the disulfide oxidative environment is there, which also means that the disulfide linkages take place properly. The SS bonds form much better. So there are a series of advantages of pushing it to the periplasm: proper folding, removal of ATG, and of course fewer proteins, so easier purification. And therefore a lot of studies have been done, not only with the signal peptide molecule itself and making better and better signal peptides. I will give you very interesting, sometimes we should talk about interesting molecular biology results. Among the attempts to make good signal peptides was this attempt to say that let us remove the codon bias in signal peptides, because finally the protein has to be made. You remember your codon bias? So I should tell you about failed experiments also sometimes. And people said, let us remove the codon bias so that signal peptide is made very efficiently. And then we will sort of have better signal peptides. And it turned out that when they removed codon bias, it actually had a negative effect. Retaining the signal peptide codon bias was actually being better. And the answer is rather complex, that you should not get the signal peptide synthesized too fast because it has to obtain some kind of secondary structure. Though it's a small peptide, it doesn't have a tertiary structure, but the signal peptide removing codon bias turned out to be a negative impact. So there are all kinds of very interesting stories in molecular biology that I don't want to tell because they are far away, but there have been attempts to make good signal peptides by mixing the structure of signal peptides, etc., etc. Even though it's a small 17, 18 amino acid long, 70 to 20 amino acid long peptide, people have tried very hard and not made more efficient peptides than nature has. So obviously we still do not understand very much. As I said, as engineers, we believe we understand when we can design. So biologists believe they understand. We should say that we don't understand, because design of signal peptides has been tried for the past couple of decades with about couple of hundred papers, and almost all of them have been only very partially successful. So the second point is that the export mechanism from the cytoplasm to the periplasm, and that's an interesting problem. And again, as I said, this is too much of molecular biology, so I will sort of quickly mention it. There are multiple secretion pathways. And I will use the word secretion to say movement from cytoplasm to periplasm, and I will use the word excretion to talk about movement from periplasm to outside. And so secretion, there are multiple pathways for secretion, including because the cell wants to sell things outside. For example, the pili have to come here and then get expressed here. So the pillin proteins are secreted. So this secretion, there are various pathways, and nowadays we talk of type 1 pathway, type 2 pathway, type 3 pathway, and so on and so forth. And there are type 1 and type 2 are for proteins, are for E. coli, which are non-pathogenic. Type 3, type 4, type 5 pathways, and I think there's also type 6, and we don't talk about them in engineering. These are for pathogenic E. coli, because these are the pathways whereby the pillin, etc. are produced. And you can see, if you have seen E. coli in the lab, you will see that hair-like pillin structures are not there. The pathogenic E. coli would always contain the pillin, because that is the pillin which allows it to attach itself to various cells in the human body, because we are talking of pathogens only with respect to us, but also other animals, and thereby that attachment allows them to form films and then allows it to infect us. So E. coli, infectious pathogenic E. coli uses these pathways. So we try to not use these pathways, though there have been suggestions that why not simply cut out these pathways and use them. After all, we are not producing pillins, but somehow there is a resistance to use the pathways of secretion which are used by pathogenic systems. Pathogenic cells also have very efficient excretion systems, but we usually do not touch it for various reasons, because we do not understand whether this process of using this secretion and excretion mechanism of pathogenic E. coli would make the cell pathogenic, because we really do not know. So we do not use these pathways, but we use type 1 and type 2. As a matter of fact, we use only type 2 nowadays, because type 1 is what is the high layer promoter, and I will not talk about it. It actually also, type 1 is also an excretion pathway. Not very efficient, because it creates, I'm messing it up because I'm trying to go too fast. It creates a HYL high layer kind of HYL high A hole in the cell by these two proteins, and allows the protein to go straight out. So the type 1 pathway is characterized by something very interesting. It is characterized by the fact that the protein gets excreted without being secreted. So one of the advantages of type 1 pathway, the high pathway, is that it actually, this is the cell, and this is the periplasmic space, and the type 1 pathway actually is a protein which sits across like this and creates a gateway. And so the protein can simply move from here right outside. So, but it is not very efficient and doesn't work for all proteins, and therefore type 1 pathway is not the preferred method. The more preferred is the type 2 pathway, which involves using which is also called the SEC pathway, the SEC A, SEC B pathway. which uses a protein called Sec-A-Sec-B, and these proteins also sit here on the extracellular space and but allow the protein to go to the periplasm. And just to sort of tell you that this is a very confusing subject and I'm trying to sort of compress it. Type 2 pathways are A and B, and A is actually Sec-A-Sec-B pathway. B is called the TAT pathway, and I'm just throwing names here because I just want you to have some flavor of the kind of complexities that are involved. Now, type 2A pathways are characterized by sending nascent polypeptides, unfolded proteins, polypeptides into the periplasm to allow them to fold. So Sec-A-Sec-B sends proteins in their unfolded state, and in the unfolded state it is sent to the periplasm where it folds. But TAT pathways send folded proteins out to the periplasm. So proteins which fold very fast in fractions of seconds cannot be exported by the type 2A pathway. And so people have tried to export small proteins because the most commonly used pathway is the type 2A pathway with Sec-A, Sec-B, and if the protein folds very fast, you just cannot export it to the periplasm. And nowadays people have realized just a few years ago that you have to use the type 2B pathway, which is the TAT pathway, which exports folded proteins. That's the only thing I want to mention in this, because otherwise I'll get into too much of trouble. And therefore the argument is for engineers. What do we do? And so we can actually co-express Sec-A, Sec-B along with the recombinant protein. So it is not a question of simply producing strong promoters. It is a question of designing the host cell. Host cell design by molecular biologists who do not know these problems. That you tell them, well, hold on, I am producing a lot of protein. Can you do a host design which has a higher expression of Sec-A and Sec-B? So it's really a question of host design. And host design which will, for example, co-expression of GRO-ESN or DNA-K, DNA-J or heat shock proteins, co-expression of Sec-A, Sec-B. These are things that ensure that the host is better designed for producing protein. So the take-home message that I'm trying to do in this single lecture is that it is not, as they will teach you in molecular biology, that making the good promoter is what ensures good protein formation. It is a design which is much more complex and much more holistic than having a good promoter. A lot of recombinant DNA technology that you will learn or would have learned would have emphasized that promoter लो, promoter के downstream में gene लगाओ और तुम्हारा काम हो गया. No, it is not done as an engineer is concerned, who is going to produce this protein in large quantities. So you have to ensure Sec-A, Sec-B are overproduced so that the type 2A pathway is active, especially for proteins which fold slowly. They go to the periplasm. That also prevents misfolding. If it is misfolding in the cytoplasm, you reduce temperature or you add GRO-ESN. And then finally, let us in the last 5 minutes. So again, I'm going to stop here. These are just flavors. In the last 5 minutes, I'm going to say that obviously as an engineer, you're interested in the protein going outside. And the best way to send proteins outside is to ensure that there are some holes in the extracellular medium. These holes are so nice that they do not damage the cell, but somehow allow the proteins to escape. And a huge amount of research has therefore also gone in. I mean, all these things that I'm talking about represent very, very large bodies of research and are very specialized topics. So just sort of mentioning this to give you a flavor. And these holes actually allow the protein to go out without damaging the cell. So what holes? How to find out these holes is the question. Now, I mean, there are many, many holes which have been discovered in the cell wall, which allows for proteins to go out efficiently without damaging the cell. And there's no point mentioning names. They will be just names to you, LPP or, UMPF or all that. So I don't want to mention these names of what. The question really is, how do you discover such things? How would you discover this? And I will give you I will try to end with a very small, quick story. And that is also important in terms of microbiology. You would have done some microbiology experiments earlier, or no? You have not done any experiments at all?
No experiments. Not any
Never.
experiments.
In any subject, you have never come to DBEB and done any experiment whatsoever?
No, never.
Oh wow, that is really tragic. But anyway, I can't help this. But anyway, you know, one of the standard experiments that biologists make you do in the lab is to look for enzymes like xylanase or cellulase, or protease or amylase production. Or I mean, there are many things, cellulase. But ऐसे enzyme producing cells ढूंढिए. And the way to search for it is simple. How do you search for it? You take a plate, you go outside into the DBEB garden, not the garden, the fields outside, or the messes which are much more dirtier and throw food, and you take the plate and you plate all the kachra, all the stuff that you have collected from soil stuff, and you take soil samples and you put them in water, pellet them so that you can remove the soil but keep the bacteria, and then you plate them, you spread it on the plate. And then this plate usually contains a starch plus medium plus LB or whatever. And then you pour iodine. And so, because the cells, there will be cells growing here. There will be cells growing here. And so in the plate, you will be seeing small cells growing everywhere from the soil samples. And because it contains a starch, and the one which has produced amylase would have degraded the starch, so the iodine will not bind. So you will see that some cells would have a clearance zone when you add iodine. And you can say that this is the cell, or this cell, these are the two cells which have produced amylase. And so this is the way you select for amylase producing cells. Then of course later on you have to improve them also. Similarly, for protease, you add skimmed milk. You add skimmed milk which contains casein. Skimmed milk because it doesn't contain fat, but skimmed milk in the plate will contain casein. Casein as the protein, milk protein. And then the protease would degrade this casein. And so you would see, I mean, I'm putting it in different color, I don't know what to draw. You will see clearance zone because the casein gives a very cloudy color to the plate, and you will see clearance zones which tells you that protease has been produced. For xylanase, you will add xylan and look for clearance zones after adding an appropriate dye which binds with xylan. Cellulase, you will add, cellulose and add, Congo red. These both are Congo red. So these are various strategies that you do to select for proteins. For example, the simplest thing to select for is antibiotic resistance. So now these are productions. But if you want to look for antibiotic resistance, you can now have a much wider field. Antibiotic resistance is very simple. Add antibiotic to the plate. With LB, add your ampicillin or plus ampicillin or whatever. And the cells which die are not resistant, but the cells which survive, now all these cells will die, but some cells here and there will come up. Some cells here, and these are by definition antibiotic resistant. So, positive selection is very easy. Where everything else which does not have your property dies. Because then in a single plate you can screen 1 million cells. But if you actually want to look for phenotype, like cellulase production or amylase production or protease production or xylanase production, then in each plate you cannot have more than 100 colonies, 10 to the power 2 cells. Because if more than 100, then they'll be saturated, and so you can't see the clearance zones. So notice that in a plate you can screen 10 to the power 2, but if you're looking for some kind of positive selection where everything else dies other than the one which is desirable cell, say antibiotic resistance, then you can screen 10 to the power 6 cells. So the question I will leave you with, which I'll answer in the next class, is how do you design clever strategies? That I want to design a clever strategy where I will like to pick out mutants which have holes which allow recombinant protein to go outside. I want to now screen, just like we screen for amylase cellulase. Now we are in a more interesting screening protocol. And this is an interesting topic that I leave with. I need a screening protocol which picks out cells which export the protein outside. That is a much more difficult question. How do you design a clever method to find out, develop a clever screen? And these are the kind of creativities that is there in good quality molecular biology. And I will leave you with this question, that how do you design a screening protocol which will allow for more efficient protein export? So I will plate everything, and the cells which export protein survive, and the cells which don't export protein die. तो फिर 1 million mutants को हम screen कर सकते हैं. This is the kind of interesting thing that biologists do and help you out in terms of designing good platforms for protein expression. I end the lecture here by essentially saying this was just to give you a flavor of molecular biology. I rushed through topics very superficial fashion, but I wanted to give you this message that protein expression is not to do only with promoters, but with much more complex host design. Any questions, or shall we stop here? So we will end this course here. I will expect you to answer this, and we will start tomorrow. There is no class. Friday we will talk about restart enzyme kinetics, unless you want any questions to be raised on this subject. This ends here, and because it ends here, I also want you people to think of when you want a quiz on this area. But this chapter is closed.
Yes, sir. We'll discuss and tell you.
हाँ, मतलब discuss and tell you that sir कभी मत करिए, सबको number दे दीजिए, ये मत करिएगा.
Okay. Yes.
Okay, thank you.
Sir, one more thing. The quiz will be conducted during class time or separate time?
Class time. It'll be class time.